All right, so mutations. So if it's going to be a harmful mutation, what would it take to go from mutation to illness? So it says from mutation to illness, just 4 steps. So first, a mutation, a change in the DNA, a change in the gene. Mutation gene codes for a non-functioning protein, usually an enzyme. So proteins are such a big category of molecules, and proteins function as enzymes. Enzymes help chemical reactions occur. So let's say an enzyme is not working, okay. The non-functioning enzyme can't catalyze the reaction it normally would, bringing it to a halt, okay. The molecule with which the enzyme would have reacted accumulates, like a blocked assembly line. The accumulation chemical causes sickness or death. So things accumulate, now they're not working properly, can cause sickness or death. So sometimes it's one mutation, sometimes it's many mutations before we have some issues. So they show an example, like people who, it says without aldehyde dehydrogenase, usually that ACE that's talking about an enzyme, is that so a person can drink one drink and get really drunk because this enzyme's not working to break down the alcohol, and the blood alcohol levels get high quickly. It says the fast flushing response kicked in after 1 or 2 alcoholic beverages. So here's the DNA. This would be the protein. It would the functioning enzyme would look like this. So the alcohol, it can fit and break it down. The non-functioning enzyme would look like this. So if there's alcohol, it doesn't fit, it can't break it down. So the alcohol builds up and then it can be toxic. So many genetic diseases, genetic, nature, DNA, that nature versus nurture thing. Many genetic diseases result from mutation that cause a gene to produce non-functioning enzyme, which in turn blocks the functioning of metabolic pathway. Now, switching to biotechnology, it says producing improvements in agriculture. If you're like, oh, I have nothing to do with biotechnology, you are wrong. When you go to the store, many of the foods that we eat have been bioengineered, meaning we have purposefully changed their DNA to suit us, to do something we want them to do, okay? So if you look, bio, we're talking about life, technology, right? So changing it to suit our needs. So genetic engineering, if you think of an engineer building things, right? But now we're doing it to the DNA. So adding, deleting, transplanting genes from one organism to another to alter the organism in useful ways, what we consider useful, okay? So how does that work? Now, I just want you to know the ideas. Of course, we can get into the details, but you don't have to get into the details. I just want you to know the ideas. So it says 5 tools of biotechnology. This is actually a pretty important slide because in one slide, it gives you the big idea, okay? So these are scissors. We use enzymes that are like molecular scissors, okay? It says chop up a DNA from a donor species that exhibits a trait of interest. For example, human insulin. We use bacteria to make human insulin. So first we have to identify the gene for coding insulin. Insulin's a protein. We cut it. Okay, that's the molecular scissors here. We cut it, amplify it. We can make a lot of it, small sample into more useful quantities, right? Then we can insert it into another species, which we've done with insulin. Insert pieces of DNA into bacterial cells or viruses, grow, okay, those bacteria or viruses, and then identify the bacteria, and then the bacteria, through transcription and translation, make human DNA, or make human insulin, which is insane. It's like, what? Can they really do that? Yes, and it's a multi-billion-dollar industry, okay, where we do this with different things. It says, okay, so this is the big idea, right? So restriction enzymes, those are the molecular scissors where we can cut a sequence that we're interested in. PCR or polymerase chain reaction. That's, again, these slides we're showing you, it's taken years and years to figure this stuff out, right, with biotechnology. So how to amplify, how to make more DNA. That's PCR, polymerase chain reaction. It's basically making DNA. Inserting DNA, right, into another species. So here for bacteria, and now the bacteria make the product we want. It says creating a gene library. Yes, we have different bacteria with different genes of interest. Identifying using a DNA probe. Okay, so biotechnology is the use of technology to modify organisms, cells, and their molecules to achieve particular practical benefits. Modern molecular methods make it possible to cut and copy DNA from one organism and deliver it into another. Yes, we do that, okay? The methods rely on naturally occurring restriction enzymes. So their enzymes are proteins for cutting DNA, polymerase chain reaction for amplifying DNA, so making a lot of it, inserting the DNA into bacterial viral vectors, cloning, identifying. So kind of this slide gives you that big overview that the other slide had a picture of. So we do this, okay, with our food that we eat. Biotechnology can improve food nutrition. So this is ancestral corn. This is modern corn. Look at the difference, okay? And that has to do with human intervention. For years and years, farmers have been doing this with plants, with what's the word? It's called artificial selection. So they breed plants that they want with the characteristics that they want. But now with biotechnology, we can actually take genes and insert them right into the plants. So yes, they've made rice that has nutrients, that golden rice. It says the addition of beta carotene producing genes to white rice has increased vitamin A content almost 25-fold. So that helps people in certain countries. Here, this is an important slide. Look at this. Genetically modified crops in the U.S. 86% of the corn that we eat is genetically modified. You're like, Miss Collins, I don't eat corn. What are you talking about? Hello. Have you heard of high fructose corn syrup that's almost added into everything? Well, guess what? A lot of it's genetically modified. So even if you tell yourself you don't eat corn, if you drink soda, if you eat certain sugary foods, bread, they add it to almost everything, okay? It's cheap. It makes things sweet and tasty, and we like it, okay? Here, cotton. So we're talking about, what do we use cotton for? Making fabrics. 93% is genetically modified. The blue are genetically modified. The green are not. Soybeans. You're like, oh no, don't go after my soybeans. Yes, 93% are genetically modified. Okay, and those are just examples of these huge crops that we use. Now, why would they do that? Why would they genetically modify them? They can genetically modify plants to be insect resistant. This is kind of a really weird one. Let's say, these caterpillars chomp on these leaves. This bacteria makes these crystals that's poisonous to the caterpillars. Well, they could take the gene out, insert it into plant DNA. Look, this is a plant cell, okay? And then the plants themselves make the crystals, that are toxic to the caterpillars. So that could be an example. Pesticide, herbicide resistance. So now they can spray herbicides on the plants, and the plants we want to grow don't die from the herbicide, but the weeds die. Okay, and you might go, oh, I think I'm a little worried. Yeah, there are definite, legitimate reasons to be fearful, okay? But if you're going to be fearful, you need to know the facts first, okay? Here, faster growing salmon. Look at this. This is genetically engineered salmon versus normal salmon. The large salmon carries a growth hormone gene that keeps it growing year round rather than summer only. So you can see the benefits. It says biotechnology has led to important improvements in agriculture by using transgenic, meaning taking genes from one organism, putting it into another, and animals that produce more nutritious food. Even more significant, reduce environmental financial costs of producing food. You have to understand, agriculture is a money-making industry, okay? It's all about the dollar, okay? Ecological health risks of such widespread use are not fully understood and potentially great. So yeah, you should be concerned. You should understand what we're using and what you're putting into your body. Or you're like, who cares? We're going to die anyway, so who cares? No, I'm being sarcastic. Okay. Fears. Oh my gosh, what the heck is this? Okay, I'm going to put it back. Okay, what was that? A chicken with no feathers, okay? So why would they do such a thing? Well, they did it because, they don't have to pluck the feathers. But when you look at it, obviously, there are issues. Can you think of the issues? Sunburn, insects, poor chicken, right? Fears and risks. Featherless birds are cheaper for farmers and consumers, but there are unintended consequences, including vulnerability to mosquitoes, other parasites. Now, here in your book, this is the concerns for genetically modified organisms, GMOs, genetically modified organisms. And some people really do have issues with this. And again, those concerns could be legitimate, right? So, concern 1, organism that we want to kill may become invincible. Yeah. 2, organisms that we don't want to kill may be killed inadvertently, and that's happened. Concern 3, genetically modified crops are not tested and regulated adequately. So are they, aren't they, and how do they affect our environment? Concern 4, eating genetically modified foods is dangerous. Maybe. What if somebody has certain food allergies, and let's say they have a peanut allergy, and let's say a certain gene is taken from one plant, put into another, and the person doesn't know they're eating those proteins from peanuts and they have some kind of allergy, or shellfish or something like that. Concern 5, loss of genetic diversity. Yes, we grow crops. Well, they're all the same. And so there's less genetic diversity. And this means nothing to you right now. But when we get into evolution, it should mean something. Concern 6, hidden costs may reduce financial advantages. So those are just some concerns, and they are legitimate concerns, I think. So more and more genetically modified foods are being created using modern methods of recombinant DNA. Among the public remain, however, numerous legitimate fears remain about the potentially catastrophic risks of these foods. So as scientists, we should be concerned. We should be ethically concerned about what we do and how it affects the environment and how it affects people. Given that their development relies on such new technology and about the long-term financial advantages they offer. Then finally, biotechnology has a potential for improving human health, criminal justice. These are sheep. What is it? These are cloned sheep, right? The cloning of animals. And we're going to talk about that in just a second. I'm almost running out of time. We'll see. The treatment of diseases, production of medicines are improved. We can prevent, cure, treat. Curing diseases is really difficult. We try, okay? More like the treating, right? The treatment of diabetes. So here, this person's injecting themselves with insulin. Again, this insulin is human. It's human insulin, but made by bacteria from the bioengineering. Yes, recombinant DNA. How about human growth hormone or erythropoietin? Yes, we can make bacteria that do this. Blood doping, athletes' performances. So using hormones, right? Let's say a person wants to get big. Well, that nature versus nurture. Nature is your DNA and how much growth hormone will you naturally produce. Nurture is working out, etc. But what if you want to get bigger than you genetically can? Well, you can take growth hormone to get bigger. Well, yes, there are synthetic growth hormones. Okay, we're going to go ahead and stop here. We just have a few more minutes to go.